its pool. Now there are several second messages which is known today: the cyclic AMP, cyclic GMP, inositol trisphosphate, and diacylglycerol, the calcium ions, and nitric oxides. Now we have learned that receptors regulate physiological and biochemical functions. In addition to that, the receptor itself are subject to regulatory and homeostatic controls, so that the receptors are subject to synthesis and degradation. Similarly, transducers and effector proteins are also regulated. In the regulation of receptors, desensitization occurs when there is continued stimulation by agonists. Desensitization is commonly described as refractoriness or downregulation. This feedback inhibition can be homologous desensitization, which is the receptor from the stimulated one, and the other one is heterologous desensitization, that is, from all receptors sharing a common signaling pathway. Homologous desensitization is directed to the receptor molecule itself. So the receptors undergo phosphorylation, proteolysis, decreased synthesis, etc. And the heterologous desensitization involves inhibition or loss of one or more downstream proteins that participate in signaling from other receptors. Now, in addition to desensitization, a receptor may also undergo supersensitization. And this is the case when we see a receptor blockade, like beta-adrenergic blockade by giving the drug beta blocker, when we suddenly stop the administration of the beta blocker in a patient with hypertension. In that case, the chronic reduction of receptor stimulation is abruptly terminated, and it may create this condition of supersensitization. So far we have learned the action of drug through the receptors. Now there are other drugs that work which is not mediated by receptors. And examples are some drugs like antacids. In antacid we see the acid-base reaction. So there is a binding to other molecules or ions. And then in mannitol we see an increased effect would create an increased volume of mannitol would increase the effect. So this is called as colligative effects. And then structural analogs of biological chemicals like some antiviral and anti-cancer agents, disinfectants and detergents and alcohol, and so on and so forth. Now the second topics of the basic principles in pharmacology is pharmacogenetics. Back in 1909, when Garrett described that in inborn error of metabolism, there is a deficiency of enzymes which is due to defective genes. And then it is suggested that the variability in drug responses is due to gene difference. And then during development, it was discovered the term ecogenetics, in which the genetic traits affecting metabolism of environmental chemical, like what happens in G6PD deficiency. So pharmacogenetics is a combination techniques of pharmacology, biochemistry, genetics, molecular biology to understand the genetic basis of response to drugs and xenobiotics. Now, what is the objective of pharmacogenetic research? The objective of pharmacogenetic research is to identify genetically controlled variation in drug metabolism and response. It is the study of the molecular mechanism causing this variation. It is the evaluation of the clinical implications and relevance of these variations, and the development of methods to identify susceptible individuals in order to prospectively avoid undesirable drug responses. This table shows us two pathways of drug metabolisms which are subject to polymorphisms. Drug metabolisms consists of two pathways, namely phase 1 of oxidation reduction pathway, which occurs in endoplasmic reticulum, and the conjugation of phase 2 in the cytosol. The acetylation pathway is a conjugation process. In here, it is depicted by the famous acetylation isoniazid. Isoniazid is a drug for tuberculosis which is inactivated by acetylsolic enzymes and acetyltransferase 2. The acetylation process is subject to polymorphisms. The Caucasians mostly consist of individuals with slow acetylator status, whereas the Asian people are mostly of rapid acetylators. Since isoniazid is an active parent drug, failure of treatment of tuberculosis may occur in rapid acetylators, whereas neurotoxicity occurs more frequently in slow acetylators. The other pathway subject to polymorphisms is oxidation reduction pathway in endoplasmic reticulum, which is catalyzed by cytochrome P450. A prototype drug here is debisoquine, which is catalyzed by the CYP2D6 isozyme. The CYP2D6 is subject to genetic variation, and it is known that the Asian people mostly of extensive metabolizer, whereas the Caucasian consists mostly of poor metabolizers. By knowing these polymorphisms, we may expect to see failure of treatment or the production of more side effects in certain individuals with genetic variation. Now, the genetic variation in pharmacogenetics may involve the whole pathway of drug metabolisms in the body, including the transport, like absorption and plasma protein binding, the transducer mechanisms in receptors, enzyme induction and inhibition, and the biotransformation itself, and through excretory mechanisms via renal or biliary. Now, how to learn the pharmacogenetics? There are several methods to study pharmacogenetics. The first is clinical observations. In this clinical observation, we have given an example of that, in which we can see a patient of tuberculosis treated with isoniazids, in which we see the rapid acetylator may have an increase in this to get the failure in treatment. Whereas in slow acetylator, we can have an increased risk in neurotoxicity. The second method is a study of the family or the twin studies. As we have known, there are two types of twins. The first one is the identical twins or the maternal twins, and the second one is the fraternal twins. Now, if we are supposed to give a drug to twins which is identical, what we can see there, the pathway of metabolisms or the pharmacokinetic profile is very similar in both siblings, whereas in identical twins, it is not similar. Now, other methods to study pharmacogenetics is through protein polymorphisms by study in animal models and DNA polymorphisms.